Great. It's my pleasure to introduce our next speaker, Dr. Ashley Parr. She is coming from the University of Pittsburgh, and she's going to bring us now into the next developmental stage, looking at adolescence and how iron in adolescence relates to different aspects of neurotransmitter levels.
Thank you, and thank you all for coming. So yeah, I'm going to jump forward in development a little bit and talk to you about adolescence. And I'll be presenting a set of studies where we've used 7 Tesla multimodal neuroimaging in order to understand the role of the developing dopamine system in driving maturation of prefrontal cortex physiology, including glutamate, GABA, and their relative balance in human adolescents. So my research focuses on adolescence because this is a period of the lifespan where individuals are acquiring novel skills and experiences that are needed to support the transition to independence into adulthood. And during this time, there's a lot of dynamic brain development that's going on, and adult trajectories are really being established. Now, initially, brain maturation and cognition proceed in this pattern of accumulation. The brain is responding to novel sensory stimuli, acquiring new cognitive abilities, and things are getting bigger. And this is really supported by factors such as synaptogenesis and the proliferation of white matter tracts earlier on. And when you hit adolescence, a lot of the architecture that you need is already there. And at this point, brain development is driven by specialization and learning. So you're getting rid of the things that you don't need, and you're increasing the effectiveness of the systems that are going to support adult levels of cognition. And I'm really interested in the role of dopaminergic development in driving plasticity across the brain that support this transition to adulthood. But the enhanced neuroplasticity that takes place in adolescence also confers risk for neuropsychiatric illnesses that emerge during this time. And a lot of these involve dysfunction within the dopamine system, within the prefrontal cortex, and neurotransmitter systems that support excitation and inhibition, and with prefrontal-dependent cognitive function. And all of these systems are simultaneously undergoing protracted maturation in adolescence. And further conferring risk, it's also a period of time for increases in sensation-seeking and exploratory drive that can be adaptive, but can also lead to risk-taking behaviors that can undermine survival. And prominent theories of neurodevelopment suggest that asynchronous maturation of dopaminergic systems that support reward processing and prefrontal systems that support cognitive control really support this peak in sensation seeking and confer risk for neuropsychiatric disorders. And so here I'm highlighting the driven dual systems model. And what's important to note here is that the prefrontal cortex, which is shown in blue, is available and it's newly matured in adolescence. However, this developmental peak in dopamine function during this time can often drive decision-making in service of obtaining rewards. And more recently, direct support for this dual systems model has been shown, including nonlinear improvements in executive function that stabilize at approximately 18 to 20 years of age, as well as adolescent peaks in risk-taking behavior. Now, although developmental trajectories in these systems have been well characterized, and a role for dopamine in reward and risk-taking, and the prefrontal cortex in cognition has also been well characterized, developmental interactions between these two systems and the neurotransmitter systems that support their development remain unclear in humans. And the dopamine system has been proposed as a plasticity system, given that we know environmental stimuli can alter its trajectory, inducing lasting neurocognitive changes into adulthood. And based largely on animal work, here I'm highlighting a recent model of critical period plasticity in the prefrontal cortex, and this was actually put forward by Bart here. And in this model, they proposed that adolescent increases in dopamine function were in fact a trigger for critical period plasticity. And developmental interactions between dopamine and prefrontal levels of GABA and glutamate support the stabilization of network function and cognitive control into adulthood. But to date, a lot of what we know about the development of these systems has come from animal models. And generally, these models support increases in various aspects of dopamine function during analogous developmental periods, including adolescent increases in dopamine concentration and receptor density. And in terms of what we understand about GABAergic and glutamatergic development in excitatory and inhibition balance, animal models have suggested that developmental refinements in EI balance that are driven by interactions between GABAergic parvalbumin interneurons and glutamatergic pyramidal neurons. And these changes in EI balance are really a hallmark of critical periods. Now, critical periods have been extensively studied in the visual cortex in early development. However, the same mechanisms are thought to be at play in critical periods in the prefrontal cortex. So at the onset of a critical period, you have these increases in excitatory neural activity driven by glutamate, and this is thought to push EI out of balance. An ongoing maturation of inhibitory GABAergic neurotransmission, as well as pruning of excitatory synapses, brings excitation inhibition back into balance to achieve synaptic homeostasis toward the end of the critical period. Now, putting these systems together, animal studies have shown that dopaminergic synapses onto prefrontal glutamate and GABA neurons increases during adolescence, as well as dopamine-dependent regulation of EI. And this regulation might be supported by adolescent increases in dopamine innervation, which play a role in shaping prefrontal circuits and calibrating EI balance. So in light of this, we sought to sort of translate a lot of these animal findings to humans to ask whether dopaminergic mechanisms similarly contributed to the development of prefrontal physiology. So in the past, our ability to examine developmental interactions between these two systems has largely been limited by the ability to simultaneously measure these neurotransmitter concentrations in vivo in developing humans. But today I'll share some novel neuroimaging methods where we've begun to address these questions. So what I'll show is that the developing dopamine system, which we've talked a little bit about today, can be assessed non-invasively using MRI, and supports the normative maturation of prefrontal glutamate and GABA levels from adolescence to adulthood. So a lot of the MRS indices of glutamate and GABA that have been obtained so far in human adolescents have been obtained at lower field strengths. And this can really limit the ability to separate the neurotransmitters that we care about from overlapping macromolecule signals. So in order to overcome these limitations, we've leveraged 7 Tesla magnetic resonance spectroscopic imaging. And here I'm going to focus on the dorsolateral prefrontal cortex, given its very well-defined role in cognition, including working memory. So we first characterized age-related changes in dorsolateral prefrontal cortex GABA and glutamate and their relative balance. And so looking at these results, what they showed was that there were age-related decreases in glutamate within the DLPFC. However, somewhat surprisingly, there were no significant age-related increases in GABA, which is what we might have expected from animal models. Now, critically, in order to understand the relative balance between these two neurotransmitter systems, we conducted a linear model that regressed glutamate onto GABA. And utilizing this method, what we see is age-related changes from adolescence to adulthood. So early in adolescence, you see this relative predominance of glutamate relative to GABA that shifts toward more balance or even higher GABA relative to GLU into adulthood. And the known functional consequences of this is increased balance. And given the age-related changes that we saw in glutamate, this shift might be driven by pruning of glutamatergic synapses. Now, in order to understand dopaminergic development, we leverage tissue iron, which provides this non-invasive measure of dopamine-related neurophysiology in the basal ganglia. So tissue iron co-localizes with dopamine vesicles, is a cofactor for tyrosine hydroxylase, which is the rate-limiting step in dopamine synthesis. And because, it's prominent in the basal ganglia, and because it's paramagnetic, it can be measured using MRI relaxometry-based techniques. So direct measures of tissue iron can be obtained via R2', which is a quantitative measure, and it reflects the difference between the effective and the irreversible relaxation rates. And using this method, recently links between R2' and PET-based indices of DTBZ that reflect dopamine availability have been shown, and this suggests that R2' reflects in part important aspects of dopaminergic function. But the primary method that I'm going to be using today is time-averaged and normalized T2 star imaging, which is an indirect measure of tissue iron as it quantifies the relative T2 star relaxation in the brain. But this method can be obtained from your echoplanar imaging data, so resting state or task-based MRI, which makes it a really scalable and accessible way to understand tissue iron in the brain. So briefly, volumes are normalized to the whole brain median, concatenated, and censored for high motion time points. And this results in one normalized T2 star value per region of interest. And here we're focusing really on the basal ganglia. Now, importantly, this measure does correlate really well with more direct measures of iron. And also using this measure, we've seen increases in T2 star-based measure of iron across adolescence within the basal ganglia. And just for today, although this is an indirect measure of iron concentration, I'll refer to it as iron for simplicity. Okay, so having characterized normative trajectories in these neurotransmitter systems, we next examined developmental interactions between tissue iron-based indices of dopaminergic mechanisms and prefrontal glutamate and GABA. So in addition to those two metrics, we also assessed working memory performance using a memory-guided saccade task. And very briefly, participants are presented with a stimulus. They're required to make a saccade toward that stimulus and then back to fixation. Following a variable delay period, they're instructed to direct their gaze to the remembered location of the stimulus. And performance is measured as accuracy in degrees of that saccade back to the remembered location. So our initial models had used generalized additive mixed models to characterize nonlinear age-related changes across neurotransmitter concentrations and working memory. And second, and perhaps the more critical analysis, we investigated interactions between tissue iron and age on prefrontal glutamate and GABA and working memory performance. And this allowed us to assess whether individual differences in dopaminergic function were associated with variation in maturational trajectories across these prefrontal systems. Okay, so as I showed before, at the population level, early adolescence is really characterized by this predominance of GLU relative to GABA, which decreases throughout the adolescent period toward more balanced GABA and GLU levels or higher GABA relative to GLU into adulthood. Now, just before describing the interactions with iron and age, I will note that the normalized T2 star-based measure is inversely related to iron content. So here, what I'm going to show you, the negative values reflect higher iron, and the positive values reflect lower iron. And in addition, these values were also adjusted for age, such that individuals who are low relative to their age cohort. So critically, we saw a significant iron by age interaction in our measure of GLU relative to GABA, suggesting that tissue iron modulates trajectories. And follow-up analyses revealed that the developmental refinements in GLU-GABA were most prominent in individuals with higher iron, shown here in the dark purple shades, relative to low iron, shown here in lighter purple. And this was corroborated with a simple slopes analysis that confirmed that the slope of age by this GLU-GABA measure was significantly different from 0, indicating significant developmental changes for individuals with relatively higher iron content. So together, this analysis suggests potential moderation of trajectories of GLU-GABA by tissue iron, again with individuals with relatively higher iron content showing the most pronounced age-related decreases in GLU-GABA, resulting in lower GLU relative to GABA in young adulthood. Now, in order to ascertain whether this dopaminergic modulation might be driven by excitatory glutamatergic neurotransmission or inhibitory GABAergic transmission, we repeated these analyses separately across glutamate and GABA. And walking through this again, glutamate significantly decreased with age at the population level. We again saw this significant interaction between age and tissue iron on GLU, and similar to the GLU-GABA measure, the developmental refinements in GLU were most prominent in individuals with higher iron, shown here in the dark blue. And importantly, we didn't see significant modulation of GABAergic trajectories by iron. So putting these pieces together, these analyses suggest that individuals with relatively higher levels of iron have the most prominent age-related changes in GLU-GABA. And this might be driven by steeper declines in GLU rather than GABA, which potentially is reflecting a greater degree of pruning of excitatory synapses in the higher tissue iron or higher dopamine group. Now, finally, we looked at the relationship between tissue iron and working memory trajectories. So first, we characterized the effect of age on our memory-guided saccade task. And as has been seen before at the group level, you can see that performance robustly improves across adolescents, with lower position error indicating better performance. We again saw that tissue iron significantly moderated the age-related trajectories in MGS performance, again with higher iron associated with a greater magnitude of age-related improvements in working memory performance, resulting in better performance in adulthood relative to low iron. And this is consistent with a role for dopamine that has been well demonstrated in working memory performance. Okay, so just to sum up, to our knowledge, this is the first study to simultaneously measure glutamate, GABA, and dopaminergic physiology in human adolescents. And this is allowing us to really begin to understand the developmental interplay of these systems in supporting cognitive function in adolescents and humans. And we've demonstrated that tissue iron properties that are linked to dopamine are playing a role in the developmental specialization of prefrontal glutamate and GABA and their relative balance, as well as cognitive function in adolescents. Now, specifically, individuals with relatively higher levels of tissue iron which is potentially in part associated with increased dopaminergic function and innervation, showed the most pronounced age-related changes in GLU relative to GABA in adulthood, and this is likely driven by steeper declines in glutamate. And this increased dopaminergic modulation in the high group might reflect an enhancement of pruning of excitatory synapses, and this is in support of animal models that have shown a role for dopamine in synaptic pruning in the cortex, particularly at glutamatergic synapses. And variation in tissue iron similarly modulated working memory trajectories, and again, this is consistent with a role for dopamine in the development of executive function. And so together, these results suggest that some of the biological regulators of critical periods that we've seen in animal models might also be playing a role in the specialization of human prefrontal cortex physiology and higher order cognition during this critical transition from adolescence to adulthood. And so with that, I'd like to thank you all for listening. Thanks again for the organizers of this symposium, as well as my collaborators and funding sources.